Yes lads, welcome to the first video of MCBHD, Applications of Molecular Biology to Medicine. Okay, bismillah, so let's start off. So we're going to start off with cytogenetics laboratory. So cytogenetics in the lab is a studying of inheritance via chromosome analysis and pre- and postnatal testing, or adult testing. So cytogenetics basically studying inheritance. So why do we do this? This may be ambiguous. There may be ambiguous genitalia or intermediate gender. So we want to identify the gender correctly. There may be known familial chromosomal rearrangement, and there may be multiple miscarriages. So ambiguous genitalia or intermediate gender, known familial chromosome rearrangement, and the family may have had multiple miscarriages. So how identifications are done. So we can split identifications into two main subclasses. As we can see, there's cytogenetics and there's molecular genetics, each in two different yellow boxes. So cytogenetics, there are 4 types of investigation we can do with cytogenetics, and these are probably aren't the only 4, but these are the 4 that we'll go into. There's G banding, FISH, qFPCR, and array CGH, and molecular genetics investigations we can do are Sanger sequencing and next generation sequencing. So let's carry on onto the structural variation in genetics. So structural variations in genetics could be chromosomal abnormalities, e.g. trisomies, translocations, and copy number variants, which are large deletions or duplications. So to review that, simply the structural variations between genes or in genetics in general are chromosomal abnormalities such as trisomies, translocations, and we should go into translocations, and if not, translocation is basically swapping of genes, and the copy number variation, which are large deletions and duplications. So scientists use 3 key features to identify chromosomes. They use the size, the banding patterns, and centrosome position. Size, banding patterns, and centrosome positions. These 3 features enable geneticists to look for key abnormalities. So, scientists use 3 key features to identify chromosomes: size, banding pattern, and centrosome position. And there are three key features that enable geneticists to look for key abnormalities. Anyway, the centrosome position is a very important way of recognizing chromosomes. There are a number of names where centrosomes can be located. So there are a number of names where centrosomes can be located. So that's, ignore that basically. There are a number of names where centrosomes can be located. So we name chromosomes differently depending on where the centrosome is located, and we can see this up here. So chromosomes with different centrosomes. So it's important to remember that this red dot is a centrosome. Okay, so a partial way to say chromosomes with different centrosomes is centrosome localizations. So there are 4 different types of centrosome localizations: metacentric, submetacentric, acrocentric, telocentric. Simple? Cool. We'll move on to the first type of investigation, called G banding. So it's the most common chromosome banding is G banding. G stands for chiasmostane. So G standing for chiasmostane, just to reiterate that. The chromosomes are taken from the metaphase part of the cell cycle. So banding occurs due to chromatin. What are chromatin? There are basically 2 types of chromatin: euchromatin and heterochromatin, each in their respective colours. So euchromatin is GC rich and is loosely packed because it's active. So euchromatin GC rich and is loosely packed because it's active. So this allows it to unwind easily. This is essential. Heterochromatin is AT rich, therefore it's tightly packed, and it's tightly packed because its genes are inactive and are not being transcribed regularly. So euchromatin GC rich, loosely packed because it's active. It's being transcribed regularly. So being loosely packed enables it to unwind and unzip easily. Whereas heterochromatin AT rich is tightly packed because its genes are inactive, so it's not being transcribed. So because of this, they stain differently with chiasma stain. It stains darkly to heterochromatin due to it being tightly packed. So as we see here, the heterochromatin stains darkly, the euchromatin stains lightly. An important point is that telomeres and centromeres are normally heterochromatin. So normally in genes we find the telomeres and the centromeres are dark. And here we can just see an example, telomeres and centromeres, normally dark. Here you can see that the telomeres actually aren't heterochromatin, but usually, and a lot of the times they are. Also go on to quickly talk about how genes are basically located in chromosomes. So they have a system, as you can see here, little code LP15. So L means chromosome 11, P means a small arm of chromosome 11, and 15 means 15 is basically the section. So 15 means this section, and the next section down would be 14. So this section would be 15, further down would be 14, 13, 12, 11, etc. And you can also go into higher resolution, which will be split by decimals. So as we see here, 11p 15.5. So we can go into high resolution by splitting this 11p15 into decimals, and that can be shown here on the picture. 11p15 and 11p15.5. We're going into further detail. We're going into higher resolution. So we start splitting them down by decimals.